this more sort of fancy model is actually doing a better job of representing the underlying structure and how it's changing over time. Now, we don't, as I said, we don't know that these things are real change points. They're places to look for more information. So if you wanted to dig into this, if this was the application you cared about, you could use these change points and the places where there's no change point as a hypothesis generation technique to say, what's going on here? Something seems to be happening right here. Let me look at what's going on there. And in particular, because the model gives you these dendrograms, you can actually ask, you can look at it and say, what changed from this point to that point? How did it rearrange the vertices in terms of how they talk to each other? So that's somewhat useful, I think. OK. So the neat thing about this is that it's interpretable. It actually tells you not just does something happen, but how it happens too. And that's quite nice. And it has these good results both on synthetic and real data. We did quantify the precision of recall for these labeled events for all 3 networks, and we found that it really does do better than those alternative techniques, which is a nice sort of validation point. OK. So last example. This is going to be a little bit different. I have to teach you some biology. Here is an ultra-condensed version of malaria 101. Malaria kills a lot of people a year, about 1 million, mostly kids, actually, because kids, their immune systems are naive. They haven't seen this disease before, and as a result, they tend to get more sick than the adults. It's caused by a Plasmodium parasite. The Plasmodium parasite is a eukaryote, so it has a very complicated genome. And in particular, there's a set of genes called the VAR genes that are critical to its pathology. So it uses these genes to build a protein. The protein, it sticks through the membrane of the red blood cell it's hiding in, and that's an anchor. It uses that to stick to the inside of a blood vessel. And it does that because if it goes through the spleen, it gets cleared. So it has to stick somewhere in order to avoid getting cleared by the spleen. But of course, when it sticks that protein out, the immune system can